David Moore is from the Baylor College of Medicine in Houston, and he's a renowned and a very powerful figure in the nuclear receptor field. I used to be his postdoc in his lab, and I don't know how he can manage it. He keeps on publishing high-impact factor papers without staying in office or in the lab. He's all the time travel around and enjoying his life. So you will see from his talk, and his lab really established the CAR as an important regulator of liver cancer. So without further ado, let's welcome David.
So thanks, Lee. Is this working? Yep. And here we go. So I'd like to start by saying, by building on something that was briefly mentioned a moment ago, which is that a very important part of sort of Cold Spring Harbor tradition worldwide now is not only the talks that the opportunity to meet and interact with people, you know, on the yard, on the green at Cold Spring Harbor, but actually also at the bar. And I want to sort of second this notion that I know that some students from this area feel a little bit shy about going in there and maybe buying a beer or something like that, but I would like to encourage all of you, again, particularly the students, to go and have a little bit of fun afterwards, right? And with that said, I'd like to also thank Jen Sheng for suggesting that I be a keynote speaker. I mean, it seems a little bit weird to me to be both an organizer and a keynote speaker, but it wasn't my idea. But I was happy to say yes, right? And I'm also happy to come back to Asia. I've actually been, of course, to China before several times, but many times also to Korea. And I took this picture over the weekend. This is in Gwangju. This is Mudongsan. This is the big mountain that is behind the city of Gwangju. And it was a great opportunity to be a tourist over the weekend and actually also to visit Korea, several meetings there. Now, what I'm going to be telling you today is really two stories, one that moves in the forward direction for CAR promoting cancer, and another one that's not as well established, but that we're really excited about, that moves in the opposite direction, targeting CAR to actually treat and prevent liver cancer, as is on the title here. So rather than giving you a lot of statistics that people in this audience don't need to hear because you've heard them already, I thought I'd try something a little bit different. Worldwide, on average, I don't know about death rates during days and nights and things like that, but on average, just the numbers say that about 100 people will die of liver cancer during tonight's session. Enough of that. We don't need to introduce liver cancer as an important topic here. So, and this is actually also probably not needed because I hope that everybody in this room is aware of the nuclear receptor superfamily that may be most famous for the receptors for steroid hormones like the estradiol receptor, the estrogen receptor, diagrammed here. And the main point of this is to be sure that everybody is up to speed by remembering that these are not cell membrane receptors. They are nuclear receptors that are bound to DNA all the time, most of them. And when they are bound to DNA, they specifically regulate target genes that are nearby, transmitting the signal of hormone binding directly to an output of gene regulation. So in some ways, they are the simplest possible signal transduction machines. We're going to be talking today about a receptor called CAR that was first described by my lab many years ago now. And CAR and its close relative PXR function in one of the many well-known functions of the liver, which is drug metabolism. So in addition to making glucose for the brain and then metabolizing, controlling bile acids and all sorts of other functions, the liver, of course, protects us against the toxicity of foreign compounds. And it does that by inducing a variety of cytochrome P450s and other drug metabolizing enzymes that are induced by the receptors CAR and PXR functioning coordinately. In response to phenobarbital and a bunch of other characteristic ligands that induce a similar response, characteristic compounds, I should say, because they're not all ligands, you see induction of a subset of P450s and an overlapping subset in response to PXR. So when the liver senses a foreign compound, either directly or indirectly, it's able to respond to that potential threat by inducing drug metabolism that clears out those foreign compounds. CAR, in contrast to what I told you just a second ago, is not on the DNA all the time. In your liver, probably 90% of people, maybe more than 90% of people, have CAR held in the cytoplasm, and all the mice in the animal care facility have it in the cytoplasm. And it's not doing anything to regulate gene expression there, of course. But in response to conventional agonist ligands, like this TC compound that we use for the mouse CAR, it translocates to the nucleus, just like as you've heard for other receptors, and of course binds to and turns on target genes. There's an indirect pathway, as I mentioned briefly a second ago, whereby phenobarbital actually does not directly bind to CAR, but instead induces a still poorly understood stress response that probably involves AMP kinase. And that results in translocation of CAR to the nucleus, where it's still able to turn on target genes, even though it doesn't have any agonist ligand. And that's where the C part of its name comes from. So it's constitutive androstenedione receptor. So it's able to turn genes on in the absence of an agonist, apparently. But it does have antagonists. And because of the quirks of pharmacology terminology, we don't call them antagonists because an antagonist needs to counteract the activity of an agonist. Instead, we call them inverse agonist formally, although I'll probably refer to these compounds as antagonists because it's simpler. So this is an experiment that we did quite a long time ago. And actually the first paper that showed that CAR was involved in drug metabolism for sure. And what it says here is that if you treat mice with phenobarbital or this TC compound that I just mentioned, what happens is that the liver increases in size. With the TC compound, it actually doubles in size, and this is only a 3-day experiment. And both of these responses are completely gone in the CAR knockout mouse. I'm not going to show you a lot of other data about this, but this is a response that's adaptive. So the liver increases in size. When the phenobarbital, for example, goes away, it shrinks back down. So this is an adaptive response. It's not some sort of bad response. It's a good response. So the liver increases its capacity to metabolize foreign compounds by growing in size. Another thing about this is that we know some things, but not that much still, about the forces that drive this, the genes that drive the proliferative response. We know that CAR induces MYC, FOXM1, polo kinase 1, and some other genes that are also proliferative in this context. Another thing about this response that's important is that it's protective in the sense of blocking DNA damage-induced apoptosis and actually other apoptotic pathways. So the liver is not only protected by inducing drug metabolism, it's actually also blocking programmed cell death. And you can see here that this DNA ladder that's induced by ultraviolet light in the wild type, this DNA ladder that's induced by ultraviolet light in the control mice is completely blocked by TC treatment, and the CAR knockout doesn't show that response. What's diagrammed here is the ability of the programmed cell death response to be suppressed totally by the TC compound. Doesn't happen in the CAR knockout. It actually also doesn't happen in the p53 knockout. This is a suppression of p53 response, and that's going to come up again later. So several years ago, Masha Negishi and my lab did the same experiment where we just asked whether the CAR knockouts are sensitive to the well-known non-genotoxic carcinogenicity, or in particular the tumor promotion by phenobarbital and other compounds. And here you can see maybe in this mouse that's treated with the DNA damaging agent DEN plus the TC compound, there are lots of tumors. And in the CAR knockout, there's no tumors. And as one or two of you will have heard me say before, this is why we call this process carcinogenesis. So more quantitatively, the CAR knockouts have no tumors at all. And in response to DEN plus PB or TC, you see a little bit of tumors here, so we never see a tumor in these mice, so they're completely resistant. So the unpublished part of this story begins with a functional synergy between CAR and beta-catenin that is important to introduce a little bit about the beta-catenin pathway. I think that people should be pretty familiar with it. It's tightly associated with human hepatocellular carcinoma, at least a subset. And under ordinary circumstances, beta-catenin is held in an inactive form in the cytoplasm. can be activated by mutations that affect phosphorylation that destabilizes it, or a variety of other potential mechanisms. But the net result of activation of beta-catenin is that it moves to the nucleus and turns on a variety of oncogenic targets and proliferative targets, including c-Myc. We got into this because of a couple of papers, actually, that were published now more than 10 years ago by Michael Schwartz in Germany. And what they found was that although most of the tumors that you get with Harvey-RAS, so most of the tumors that you induce without phenobarbital, if you just do DEN alone, the most prevalent mutation that they found in this early stage was Harvey-RAS activation, 6 out of 20. And none of the 32 tumors that came with phenobarbital had activating mutations in Harvey-RAS. But in contrast, the mutations in the tumors that were induced by phenobarbital, almost all were associated with beta-catenin. So 37 out of 46, so nearly all of the tumors that you induce with phenobarbital have activating mutations in beta-catenin, which of course immediately suggested a direct interaction of some sort between CAR and beta-catenin. So we looked pretty carefully for the sort of simple things. Beta-catenin has actually been described as a coactivator for other nuclear receptors. We didn't see any evidence for that, and CAR activation didn't seem to affect beta-catenin transactivation either. But we decided that we would pursue this in more detail. And we had a specific hypothesis that relates to the fact that CAR induces that pattern of liver growth that I just told you about, but that process is somehow self-limited. The liver doubles in size and then it stops. As we found later, and was actually also partially described, what beta-catenin can also do a similar sort of thing that's also blocked. So what we thought that the interaction between them might be that beta-catenin was able to overcome the blocked CAR-induced growth and vice versa. It looks a little bit complicated, but it's actually pretty simple, right? And we used a model that has a floxed exon 3 that removes the phosphosites that destabilize beta-catenin. So this is actually, this loss of this exon 3 is an activating mutation gain of function. And when we did this initially with a heterozygous floxed allele, what we found was that in the mice that had just activation of CAR, you see CAR targets like 2B10 and MYC going up. CRE doesn't, which activates the beta-catenin, doesn't induce this. It actually didn't induce c-MYC or cyclin D1 in this experiment either, but it does induce glutamine synthase, and this is the combined activation. So we didn't actually see what we predicted that we might see there, but we were, the person who was working on this, Bingning Dong, was very patient. And what she found was that rather than the acute growth that we were anticipating, what we saw was essentially a recreation of the results from the Schwartz lab in a different context where if you pre-induce the mutation in beta-catenin and you pharmacologically activate CAR, you see essentially the same result, which is tumors, right? So we saw tumor genesis. So these pathways in combination are sufficient to induce tumors in mice without any other agents. In collaboration with Jusog Lee at MD Anderson, we actually profiled the RNA from these tumors. And what we found was that, it's maybe a little bit hard to see, but this group of human tumors here matches the mouse tumors here. And it is the subset of human tumors that have activating mutations in beta-catenin. So although it was done in an unbiased way, what we identified was a very tight correlation between this gene signature that's identified in both the human and the mouse tumors. Initially, to my surprise, but in retrospect, not, the people, the humans that have this signature, and the beta-catenin signature, as Jusog called it, actually have better survival, better long-term risk-free relapse-free survival and overall survival. And this is correlated with the fact that beta-catenin mutations are associated more with adenomas than with later stages of cancer, liver cancer. So when we did this experiment in the homozygous mice, what we found was that we were able to induce not only glutamine synthase with CRE alone, but actually also cyclin D1, and we were able to increase liver size. And you see a little bit of an increase when both of them are activated together. When we waited a little bit, what we found was that the block to CAR and beta-catenin-induced growth actually was overcome in the doubly activated mice. And this is a sort of picture worth 1,000 words experiment. So this is the control mouse here, and this is the liver of the doubly activated mouse. At this point, it's about 25% of the body weight of the mice. And it keeps growing until they die. We believe that they die of respiratory insufficiency due to pressure against the diaphragm. We're not sure about that. But at this point, it's important to emphasize that the liver appears to be pretty normal. So serum parameters of liver function are all within pretty much normal range. Maybe a little bit of an increase in ALT, as it says under the clock here. But bile acids and glucose homeostasis appear to be normal. So it's a relatively normal hepatocyte function that's maintained despite the very dramatic increase in liver size. So just briefly, we know something about what happens to one half of that double arm thing, which is, as I've already told you just very briefly early on, CAR induces MDM2. So that's relevant here because the pathway of beta-catenin activation was known in other tissues, and we confirmed in liver, to be associated with a response that's called oncogene-induced senescence. And in this pathway, what happens is that the cell senses that there's something wrong with its proliferative pathways, and rather than inducing cell death, it actually induces senescence via activation of p53, typically, which induces, can induce cell death, but also can induce senescence via p21, as is diagrammed here. So p53 activation induces p21. That's what the circle is here. Of course, you know that p53 actually also activates MDM2, which negatively regulates p53 and induces its turnover. So in this case, there's a sort of a trick because both pathways are inducing MDM2, but the CAR activation is selective and specific for MDM2, and it occurs prior to the senescence. So what happens is that the CAR activation completely overcomes the senescence. Yeah, I didn't think that you would be able to see. Maybe someone in the front can see that there's some blue dots here, but quantitated down here, there's a very clear induction of senescence in response to the adenoviral Cre, in response to beta-catenin activation, and this is completely reversed by CAR activation. You see the same thing with inflammatory markers that are associated with senescence. And here's the MDM2 pattern where you can see that CAR and p53, CAR and beta-catenin both induce MDM2. In this case, without the brake being on beforehand, what happens is that p21 is activated. But when the brake is on beforehand here in the double activated, p21 is unable to accumulate. So the pre-existing MDM2 basically eats up the p53 response once it occurs. And so at the level of protein, there's no p21 here, but you can see clear protein here. p53 is activated here and not in the doubly activated. The last experiment along this line is that the results I just told you about were done with high titer adenoviral Cre expression. And you might imagine that a low titer adenoviral Cre that sort of infects only a few foci of cells would result in preneoplastic lesions that would then progress to liver cancer. And sure enough, that's actually what happens. So this is another way of saying that CAR and beta-catenin are sufficient to induce tumorigenesis. So with the remaining time, I'm going to focus on some what I find really exciting results that are still at the relatively preliminary stage that really don't have complete mechanism, but do have, as you'll see, some pretty striking biological responses. So even before we knew that CAR was involved in drug metabolism, in collaboration with Ron Evans' lab, we identified the first inverse agonist ligand for CAR, which is the androgen metabolite called androstenol. And if you activate gal-mCAR, so just the ligand binding domain of mCAR, or in this case gal-mCAR with RXR, so it's able to, so gal-mCAR plus RXR is sufficient to activate target genes. As I said, you don't need to add anything else. It activates it all by itself. Right? But when you add androstenol, what you see is actually repression of this transactivation, and it goes to a level that's below the level of the control. So it appears to convert CAR from an activator to a repressor. And we and others have found evidence for interaction with co-repressor in this context here. Now, something that I didn't tell you yet, but I'll tell you now, is that both CAR and PXR are much more variable than other members of the nuclear receptor superfamily. So mouse thyroid hormone receptor and human thyroid hormone receptor are virtually identical, and of course the hormones are identical. The same thing is true for most of the steroid hormones and things like that. So the receptors are tightly conserved, and of course the hormones are conserved. Sometimes there's a difference, cortisol versus cortisone. But The xenobiotic receptors, CAR and PXR, are more variable than other receptors. This actually correlates very well with quite distinct biological profiles of xenobiotic response of rodents and humans. This is a big problem for the pharmaceutical industry because they can't test the toxicity of their drugs in rats and mice and expect to see results that will be the same as what they see in humans. So in an experiment later on, we screened basically a small library of compounds that were already on the market and things like that, a small library of drugs and other compounds. And what we found was 2 hits for compounds that would shut off human CAR as opposed to mouse CAR. This compound here, CITCO, is a distinct agonist for human CAR, and sure enough, it activates human CAR here. And you can see here that both meclizine and clotrimazole are able to inactivate human CAR and appear to be inverse agonists. There's a recent paper from another group that questions whether this is for real, and I don't know whether they are right or we are right, but clotrimazole, and there are actually other compounds that appear to act as human CAR inverse agonists. So this is a clotrimazole result. It's a little bit complicated. What we have here is a cell line that's actually a HepG2 derivative, so human cell line, that is expressing either mouse or human CAR under the control of a tetracycline-inducible driver. And so you have no expression in the control. If you induce with a low level of DOX or a higher level of DOX, what we're looking at here is the UGT1A1 gene. So this is one of many genes in drug metabolism that's a primary target for CAR, both human and mouse. And again, if you activate with DOX at lower or higher dose, what you see is the apparently constitutive activity of CAR driving expression of the endogenous human UGT1A1 gene. The CITCO compound that I just introduced on the previous slide increases expression above this constitutive level. And you see, in this case, a little bit of a bump also with the TC compound. Different genes, so different responses. Some of them are induced much more strongly by the agonist, and others are mostly all the way there without it. In both cases, what you can see in the presence of this level of doxycycline, or this level of doxycycline, is that, in this case, clotrimazole for human CAR or androstenol for mouse CAR really drives the expression way back down, all the way back down. So it converts this, it basically inactivates this transactivation. So clotrimazole here, androstenol here. What's diagrammed here, this is a ChIP-seq result. So this is, we identified all of the binding sites for human and mouse CAR in these human cells. And this is the UGT1A1 gene. And you can see here that these are the different binding sites for human CAR near the UGT1A1 gene. And the point that I have for the reason that I'm showing you this result is that this doesn't happen because CAR falls off. It happens because the CAR is still there but unable to transactivate, and possibly also active repressor. So this result reinforces the ability of CAR to really shut things, of the inverse agonists or antagonists to shut things off. So people in this audience should be pretty familiar with the concept of tamoxifen treatment of breast cancer, or of course there are analogous results with antagonists or just ablation of androgen presence in prostate cancer. In this case, Kent Osborne, who is now the director of the Cancer Center at Baylor, was showing that you could treat, this is an early paper showing that you could actually treat women with breast cancer with tamoxifen, the estrogen receptor antagonist. As you probably know, this is effective for a modest period of time, and then typically the tumors become resistant. But it is a mainstream treatment of breast cancer treatment therapy today. And of course, there are other more modern antagonists. This is also an interesting part of the story that is a relatively early result that's since been borne out by other studies. Basically, in women that are taking tamoxifen for hormone replacement reasons, the incidence of breast cancer is down by about half. So very significant decrease in the incidence of breast cancer in women taking tamoxifen. So it not only treats very clearly, but also prevents breast cancer. So, of course, we wondered whether CAR would basically be the same thing for liver cancer that ER is for breast cancer. And in this early experiment from a while ago now, what we did was just take primary hepatocytes and treat them with a very potent mitogen, namely epidermal growth factor. And we co-treated them with or without androstenol. And I think that you can see here, just by BRDU staining, that there's a lot of proliferation in the, of course, mitogen-treated primary hepatocytes. And androstenol doesn't bring it down all the way, but it's well below half, right? So it potently suppresses the induction of proliferation by EGF, exogenous. So this experiment, I think, is important because it is, so we're not talking about phenobarbital here. We're not, mixing phenobarbital and androstenol here. We're talking about an exogenous, heterologous sort of mitogen, but nonetheless, androstenol is able to overcome that stimulus, at least in part. This is another early experiment where we actually did combine phenobarbital and androstenol. And we did the standard DEN plus phenobarbital treatment, where we wait, you treat with DEN, you put constant treatment with phenobarbital for 8 months. At this point, they have tumors. We started to treat with or without androstenol by daily injection for 2 weeks. And what you can see here is that in this early experiment, the number of tumors per liver appeared to go down pretty significantly. And the tumor weight, I should say the overall liver weight also went down. So this experiment was exciting, but it's also a little bit frustrating because you can't know what happened to these tumors when they were here, and then you're only looking at them later on. So we only saw the outcome, right? So we took an alternative approach, and I'm going to be telling you several different versions of this story. In the first version, we started with wild-type ICR mice, which are more sensitive to liver tumors. And we treated them with DEN alone. So this is a standard regime where you treat them at an early age when the liver is still proliferating. It's a standard, sort of genotoxic tumorigenesis. And again, we wait 8 months. And now we image the mice with MRI at that time point. We treated them with solid time-release pellets that release androstenol. And we waited 3 more weeks, and then we measured them again. And in this experiment, the wild-type tumors didn't really grow very much. They were established, but I don't know if you can see them very well. They were established, but they didn't increase in size that much. But the androstenol-treated tumors decreased in size dramatically by more than half. So over this 3-week experiment, which actually reproduced an earlier experiment in a different model, what we saw was about 50% or more decrease in tumor size. We're still working on the details of the mechanism. As I told you, we don't really have a clear sense of how CAR drives proliferation, or even for that matter, all of the pathways that drive tumorigenesis, that drive proliferation of these liver tumors. But I hope that you can see that there are some brown spots here in the wild-type tumors that are treated with androstenol, and you don't see those in the you don't see any apoptosis in the untreated tumors. There's actually also, of course, quite a lot of proliferation by Ki67, and I'm not showing you that in the tumors. And the proliferative index is actually very dramatically decreased also by androstenol. So it stops proliferation and does induce apoptosis, which more or less had to be the case if you're going to be shrinking so much in size. We're pursuing the question of what the targets are. We have a little bit of information. I already mentioned that polo-like kinase 1 and FOXM1 are induced by CAR activation. Also c-MYC. This is an experiment in just normal livers and you would see something similar with c-MYC, where you see a pretty robust induction in response to the TC compound in this short-term treatment. And of course, the CAR knockout doesn't show any response to this, right? So these are CAR-dependent pathways. We don't have much evidence for primary response of either FOX or c-MYC, FOXM1 or c-MYC. It looks like there may be some indirect pathways there, but PLK1 does have a very clear binding site by that genome-wide analysis that I showed you just a minute ago. Both FOXM1 and PLK1, although not so much c-MYC, do go down in the tumors. They're up in the tumor tissue, right? This is now at 8 months later when the tumors are established. FOXM1 is induced. PLK1 is actually also very strongly induced. And if you treat the tumors with androstenol, what you see is a pretty significant decrease in both of these proliferative targets. So this is consistent with the decrease in Ki67 and actually potentially also with the increase in tunnel staining. Now, this story is a lot more complicated, and I'm just going to show you one slide here, We've done gene arrays. I think we need to do more gene arrays to get a better handle on this because the tumors are somewhat variable. But what we see is at least the indication of an increase in fatty acid oxidation in response to androstenol treatment. And also, lipin-1 is a lipogenic factor that's both a transcription co-regulator and an enzyme. And so there's evidence in the gene arrays, and we've gone on to confirm this in several different ways, that the CAR pathways may actually also be involved in metabolic regulation. What we think, and I'm not going to show you, as I said, any more details, what we think is that it's now become popular in the world of tumor metabolism to, for people to realize, the Warburg effect is not a mystery after all, right? You need the Warburg effect to allow carbon substrates to just build the cells, right? You don't want to waste all that carbon using, making ATP. You want to use the carbon to actually grow the cell. And along that line, you probably know that metformin has anti-cancer effects and things like that. So the anabolic pathways that the cell needs to build things up are, of course, counteracted by, just burning things up, right? So if you're inhibiting lipogenesis and promoting fatty acid oxidation, you may well be blocking the availability of lipid building blocks for cell growth. And we have some evidence in favor of that, like I say, but I'm not going to. We're not all the way there yet. But that's what we're thinking. So 2 more experimental results in a different system. In this case, we're using transgenic mice that are called ATX. They have the hepatitis B X antigen is a transgene. They were made and studied by Betty Slagle at Baylor. And they have some clear differences to wild-type mice, but one of the differences is actually not spontaneous tumor genesis. So despite the fact that they express a functional hepatitis B X antigen, they don't have a higher level of spontaneous tumors. And they actually also don't have a higher level of tumors when you treat them with DEN. In experiments that I'm not going to show you, they actually do have a higher level of tumors if you treat them with TC alone. So CAR does have some functional synergy with hepatitis B X antigen. But what's more important here is it's basically another version of exactly the same model in this case. And what we're looking at is the ability of the tumors that are formed in the DEN alone treated ATX mice to respond to androstenol in exactly the same way. Here we actually saw what appeared to be a decrease, a pretty significant decrease in the number of tumors that we could count. And what we saw in this case was not a shrinkage of size, but rather a block to the proliferative response. So there's a very potent increase in tumor size, 700% in the wild-type mice here in this experiment. That was blocked completely in the androstenol-treated. When we did a longer version of this experiment that was actually 2 months, we did see, we started it actually a little bit earlier, and the proliferative rate of the ATX tumors was less, and the tumors actually did shrink in size. So in this aggressive case here, the tumors actually stop proliferating, and they may actually also reverse if you treat them longer. So the last experiment that I'm going to show you, and we see basically the same thing with blocking Ki67 and inducing apoptosis. So the last experiment is a prevention experiment, and in this case, we treated them with DEN in the same way. And then we waited only 2.5 months. And we began to treat, we treated them with androstenol pellets. We actually take them out after a month and replace them. And after 4.5 months, there are a number of small tumors are present in the placebo-treated mice. If you look in detail here, you can see that some of these mice have none, some have 1. Here's a guy that has 3. But there's a very strong inhibition of tumor genesis in these mice that are treated over this relatively short time span with androstenol. But I think that this is clear evidence for preventative effect. So what I tell the students in my lab, and now I have an opportunity to sort of say it in a somewhat different context, someday everybody in China is going to be taking meclizine. Maybe in southern China, areas where people who have been infected with hepatitis B virus. I don't mean to be, make a joke of it. But I think that it may actually have utility not only in the treatment, but maybe also potentially even more utility in the prevention, meclizine or clotrimazole. So this brings me to the end and an important question, which is, as some of you will know, the people in the pharmacology world don't believe in this kind of stuff. And the reason is that if you treat patients with phenobarbital, if you look at epidemiology of human patients treated with phenobarbital, you don't see an increase in hepatocellular carcinoma. There are reports in the literature, my patient was taking phenobarbital, and sure enough, they have HCC, but in the population as a whole, it looks pretty clear, although there's some counter-arguments. It looks pretty clear like phenobarbital does not induce tumors by itself. So I was at a meeting a year and a half ago in Germany that was trying to address the question of whether these results are, whether this is relevant to human biology or not. And I made the case that I believe very strongly that it is, and there are several reasons for that. One of them is a result that I showed you today, which is that the tumors that we get in mice really do match very tightly to at least a subset of human hepatocellular carcinoma. We'll be hearing later on in the meeting and probably in the next talk about other subsets of hepatocellular carcinoma that have different properties. But the beta-catenin set matches the mouse tumors that we make. Of course, it goes without saying that we are exposed to genotoxic agents. And we have evidence in collaboration with Dimi Bissig, a result that I didn't show you today, but CAR does induce proliferation in human hepatocytes that are growing in mice, and that's an important result that we're still confirming. But finally, there's a last point that I want to make, which is the reason that these guys, basically, it's weird to me because they have completely misinterpreted their own results. Because in mice, the biology is very clear. So you get a few tumors with DEN treatment. If you give the mice phenobarbital afterwards, you get way more tumors, lots more tumors, right? So it's phenobarbital driven, and DEN must be there first. If you actually do it in the opposite direction, if you give phenobarbital first and then give them DEN, they don't get any tumors. That's because you induce drug metabolism, right? So if you translate the biology of the mouse system to the human system, what you expect is exactly this, right? Because what you expect is that phenobarbital should actually protect human patients against liver cancer. It shouldn't induce tumors. It should actually protect against tumors. The only people that you expect to have a phenobarbital induction of tumors are those that were just exposed to a DNA-damaging agent in the recent past or something like that. And so presumably that population is small, which is why this is the outcome that you actually expect. So I don't know whether they really bought my argument or not. I don't know whether you'll buy my argument or not. But I think that it's a kind of a strange thing that the epidemiologists have completely misunderstood their own problem, in my view. So what I showed you today is that full activation of CAR plus beta-catenin is sufficient to drive uncontrolled hepatocyte proliferation. We're actually trying to exploit this to grow unlimited numbers of hepatocytes in a dish, but we haven't gotten there yet. CAR acts synergistically with beta-catenin to promote hepatocarcinogenesis. I think that was already pretty clear from the previous results from the Schwartz lab, but now is also even more clear with our own results. And finally, the preliminary, but I hope you think exciting results that I showed you at the very end really do establish in my mind that human CAR is a potential target for hepatocellular carcinoma treatment and potentially also prevention. And if I had hepatocellular carcinoma, I would be taking meclizine and clotrimazole. And if any of your patients are interested, if you have patients who you think might be interested, I've tried to convince a couple of hepatologists in the U.S. This might be worth a shot on compassionate use basis, but I haven't convinced anybody yet. But if anybody is interested in talking to me about this, I would be excited about that. So Wendong and June, coincidentally, actually it is coincidence, but so there's an entirely Chinese population here that is driving this project. Wendong and June were a postdoc and graduate student in the lab that did the early studies with the CAR knockout and phenobarbital. This is Bingning Dong, who has really driven most of this project. And Ray did the genome-wide binding analysis that I told you about. Nan has joined on, and she actually did most of the ATX experiments along with Bing. And Ying is a graduate student in the lab who's actually looking not at CAR as a target in liver cancer, but CAR as a target in other potential cancers where it actually is ectopically expressed in a small number of other tumors. And with that, it looks like we are a couple of minutes over, and I'd be glad to take any questions. Graduate students first, right? and postdocs.
Very interesting work. I'm wondering the CI inverse agonist just reduce the tumor proliferation or cause the senescence of tumor cells in the later stage of HCC.
Right, so we need to do more studies in the longer term to actually see how far we can press things, how far we can drive things. But it definitely induces apoptosis. We can definitely see programmed cell death by the tunnel assay in the androstenol-treated tumors. So I think it's beyond senescence. They're actually driving cell death. It's not really clear why the cells are dying, but they certainly are. And we're far from the stage of being able to say it will, bring them, you can treat them effectively for 2 months, and then resistant tumors begin to come back, where we haven't any evidence for that sort of thing yet.
The second question is, okay, what's the relationship between CR and beta-catenin mutation? Beta-catenin mutation, do they regulate the same downstream targets?
Right. So in terms of the relationship in the original results, so in the original tumors that were studied by the Schwartz lab, the beta-catenin mutations were spontaneous, and they were apparently strongly selected for. So there's a strong functional selection for that interaction. In terms of regulating downstream targets, they do regulate some common targets, including both MYC and cyclin D1. And the reviewers keep asking us about this, we don't see much evidence for direct functional interaction. We see, in fact, no evidence that they interact with each other physically. We see no evidence they affect each other's transactivation. They do regulate common target genes, but not necessarily by potentiating each other's activity. We think that the functional interaction is actually a deeper one, that for example, as I told you, we believe that the pathway of induction of MDM2 by CAR overcomes the senescence pathway program for beta-catenin induced senescence. And so that's not an interaction at the level of individual gene promoters or protein-protein. This is an interaction between the different programs that are induced by the two transcription factors.
Okay. I have a third question. You use the CR agonist to stimulate the primary human hepatocyte to proliferation. I wonder what's the mechanism the CR can promote the primary hepatocyte to proliferate, because in our lab the primary hepatocyte is difficult to proliferate.
Right. So so in many studies of primary human hepatocytes, particularly in treatments with phenobarbital, so phenobarbital does induce proliferation of mouse hepatocytes, mouse primary hepatocytes, but apparently does not induce proliferation of human primary hepatocytes. We actually haven't done those experiments ourselves. And as I briefly alluded to, this experiment with our human hepatocytes, they weren't in a dish, they were in a mouse. So they were a humanized mouse model that Demi Bissig has, which is analogous to one that Marcus Grompe has, in which primary human hepatocytes actually replace the mouse hepatocytes in the liver. And so we're looking at a more in vivo sort of situation.
Okay, thank you.
So the in vivo relevance of the phenobarbital CAR issue is of course an important question, and this epidemiologic data is perplexing.
To me, it's actually not perplexing at all.
Yeah, I know, but if your theory is correct, then the people that are taking phenobarbital should have less mutations in the beta-catenin pathway.
I think they should be protected.
Have you looked at that?
No, that's a good point. We haven't looked at that. There are a lot of, right, so I agree that it is, perplexing, yes, but and I guess the perplexing, so there are lots of predictions. There are lots of interesting things that you might imagine, but they're way outside of our ability to do, to really do so. And many of these studies are done. There's an important one from Kaiser Permanente that's 10 years ago or more than that, and so it's really very difficult, I think, to go back and do some of the things, next round of investigation that you might want to do. But I agree that there are lots of predictions that you might have.
Hi, David. Do you think there might be some crosstalk between CAR and PPAR alpha?
That's an interesting question. So PPAR alpha is another one where the idea is that the rodent tumorigenesis results are not considered relevant to the human studies, right? And when we haven't looked at this very much at all, but we did do some liver growth studies with fibrates, and we saw, if anything, even stronger liver growth in the mouse models, which is consistent with published reports, right? So whether those pathways are overlapping, I don't know. And it's an interesting question whether there would be functional synergism between PPAR alpha and beta-catenin. I don't think there's anything known about that.
Thanks. So David, this is a pretty interesting presentation.
Thank you.
So, you know, in children's tumors, liver tumors, where you have actually mutated beta-catenin and upregulation of MYC, do you know anything about the role of CAR in this system at all?
So Milton Feingold is at Baylor, as you know, and we've looked a little bit with Milton to see if we could find evidence for that. And that brings up another important point. So that on the list of the signature that I showed you, there are a bunch of genes that are beta-catenin targets. There are also a bunch of genes that are CAR targets, right? But so that suggests that CAR may be activated in the human liver cancer. It's expressed at least at the messenger RNA level, and we have some evidence at the protein level in most hepatocellular carcinoma. Right? But when you look for some of our favorite CAR targets like 2B6 in the human, you don't see that going up. So the issue of whether CAR is really activated is not as straightforward to answer as we would like. So we don't have direct evidence, particularly, so you're right that beta-catenin is particularly associated with hepatoblastomas. Mm-hmm. And that would be a great place to look, but we haven't been able to get much evidence for that.
But have you done your experiment in the p53 null mice?
We have not done the p53 null, no.